Hello everybody, welcome to my video. My name is Shahoon Khan, and today we're going to be discussing my summer project on TOR signaling and how it alters mitochondrial activity to promote growth and development. The mitochondria are organelles that are classically known for their role in ATP production, which is its classic bioenergetic function. However, the mitochondria are also implicated in many other pathways such as biosynthetic function, cell signaling, and apoptosis. Overall, the mitochondria will work to promote growth and survival. Today, we're going to be focusing on the metabolic aspects of the mitochondria. So over on the right, we have bioenergetic function, which is the classic function of mitochondria that takes fuel molecules and uses them to generate ATP. Conversely, we have biosynthetic function, which is taking those same fuel molecules and instead of using them for ATP production, we're going to use them to increase cell mass and overall promote growth and development. So my project specifically focused on how the target of rapamycin or TOR pathway alters this dynamic equilibrium. To study this, we used Drosophila larvae, and the reason why we use this model organism is because in the larval stages, we see this exponential growth, which occurs over a span of 3 to 5 days, making them an excellent model organism to study growth and development. So previously in the lab, we have shown that increased TOR signaling is inversely correlated to bioenergetic activity. And this is seen through examining the mitochondrial ultrastructure, where as the larvae develops, we see the ultrastructure becomes denser and denser, which indicates increasing bioenergetic activity. So how might TOR be altering the mitochondrial metabolism? We suspect that it is doing so through the mitochondrial transcription factor A, known as TFAM. Now, TFAM is a protein that goes into the mitochondrial DNA and increases the expression of oxidative phosphorylated complexes that make up the electron transport chain. This suggests that increased TFAM activity will increase bioenergetic function. So how will we study TOR, TFAM, and the implications it has on mitochondrial metabolism? We set up a very simple experiment where we took Drosophila larvae 70 hours after egg laying, and we transferred them to either high nutrient food or low nutrient food. And we then examined how long it took for them to reach pupariation. We also knocked down the specific proteins that were of interest, such as TOR and TFAM, in the fat body, and we examined the effects it had on growth and development. So when we knock down TFAM specifically in the fat body, we see this accelerated developmental timing. And this is seen for both the normal and low nutrient conditions. Next, we want to see how activating TOR signaling affects developmental timing. And we did so by exploiting this upstream positive regulator of TOR. So we decided to overexpress Reb, which will increase TOR signaling. And when we overexpress Reb in the fat body, we see this accelerated growth. And this is seen for both normal and low nutrient conditions. Next, we want to see what would happen if we knock down TFAM and pair this with inhibition of TOR. And we inhibited TOR using either rapamycin, the classic TOR inhibitor, or we exploited this S6K protein, which is a downstream positive effector of TOR. So when we knock down S6K, we decrease TOR signaling. So when we knock down TFAM, we see the accelerated development. But when we knock down S6K in the fat body, we see this delay in growth. And now to our surprise, when we pair TFAM knockdown and S6K knockdown, we see this rescue in the growth. And this is seen for the rapamycin trial as well, where TFAM accelerates growth, rapamycin delays the growth, and then TFAM paired with rapamycin accelerates our growth. So all our results pretty much piece together to form this puzzle and allows us to propose our model. So in the presence of nutrients, TOR signaling is upregulated. And when TOR is upregulated, it inhibits the TFAM protein. And when TFAM is inhibited, we see this switch from bioenergetic metabolism towards biosynthetic metabolism, which is going to promote faster systemic growth and development. On the other hand, under low nutrient conditions, TOR is inhibited. And this allows TFAM to push the mitochondrial metabolism towards a more bioenergetic function, which is going to slow systemic growth and development. Now, I would like to take a chance to acknowledge Dr. Graywall for having me in the lab, and I would like to thank the rest of my lab members. And I'd also like to give a huge shout out to our funding sources. And lastly, I'd like to thank you guys all for watching.